If you're a microbe that's going to be eaten, how do you avoid being eaten? That's the next part of this. The first one is the mechanical thing, that we have this size too small or too large. And this is part of the reason that bacteria are so successful in the world, that they're small enough, they're difficult to filter out, right? They're basically making a living by being super efficient, super small, super hard to eat. Or you can get to be too large. So we've talked about this in a number of different contexts. We've talked about, say, Daphnia having large spines in the presence of predators, or algal species that make themselves long chains, like Melisira. We had the example of Melisira and sinking rates, right? So there were three things that went into that Melisira and the sinking rate. It's a planktonic diatom. It can make itself long and skinny, and therefore it has a lower, there's more viscosity of the water, so it has a lower sinking rate. It maintains the same surface area to volume ratio because it's a cylinder, which does that as it increases, so it can maintain its nutrient uptake rates. But then the other thing is that when it runs into Daphnia, it clogs the filter part, so Daphnia rejects it and puts it out, right, or any other filter feeding zooplankton. So there's those sort of selective pressures for being large. Now, if you remember what it looked like when we went to the samples in Pot State Lake number 2, there was a lot of filtering zooplankton in there, and if we saw any algae, it was generally pretty big. And we see things like serratium, which is the dinoflagellate that has the big spines on it, or long chains of Melisira, or things that are difficult for organisms of that size to handle. Then there's the feedback of, there's a lot of fish in there eating the zooplankton, so the zooplankton can only be so big. So the zooplankton that are big enough to really handle big stuff aren't there. And the algae that is able to get by that produces these spines. And sometimes, because these cost energy, the species will actually only put out the spines in response to chemical cues that they detect that are released into the water, either from the grazer itself, or if their cell contents, if there's broken cells of their conspecifics. So if something's out there eating and breaking cells of an alga of the same species, like, man, my genetic line is being eaten, time to grow spines. It's behavioral to escape, and there's actually evidence that some species of microbes will go away from compounds that are released by organisms that are harmful to them in one way or the other. So they can use, essentially reverse the random walk model, and if there's something that might harm them, work their way away from it over time. Likewise, we talked about those magnetotactic bacteria that have taxis that gets them down the sediment. Well, that puts them down in an environment where they can grow better, but also they're avoiding being eaten by the planktonic organisms that are up in the higher in the water column. And zooplankton are on the edge of being considered microbes. I mean, they're multicellular, but they are microscopic. They actually will make swarms. If they see a predator coming, they will try to get away from it. So we had an example of the Van Dorn bottle, right? The Van Dorn bottle, we said, why is that Van Dorn bottle clear? Well, it's because of zooplankton, if it sees a large shadowy object, it gets out of there. And it turns out that you can catch less zooplankton if you use a colored object versus a clear object to catch them, whether it's Schindler trap or the Van Dorn bottle. So once you get big enough to have an eye spot and sort of get an idea of what direction is, then you can run away from stuff behaviorally as well. So we talked about trait-based versus density-mediated competition. And the idea in competition is this idea of exploitation versus interference competition. So essentially you can compete in two ways. One is that you can just use it faster. You can exploit the resource faster than your neighbor, right? Be better at taking up nutrients than your neighbor. The other one would be to say, okay, I'm going to put out a compound that's going to poison my neighbor, so it'll slow him down, right? That would be interference competition. So there's a lot of allelochemicals, chemicals that are released by microbes that influence the behavior, or the activities of other organisms. And most microbial interactions, probably the majority of microbial interactions, are actually dominated by chemically mediated interactions. And then we talked about resource ratio theory already, and I can go back over that again. This is an exploitation competition. So if you remember this, we have growth rate in silicon on this first graph on the X concentration, the growth rate on the Y. And we had a species 1 that did well at high concentrations, and a species 2 that did better at low concentrations. And then we have another nutrient such as phosphate on the X-axis, growth rate on the on the Y-axis, and this species 1 does better at low phosphorus concentrations and not as well at high concentrations. And so when we have this situation, if you have high silicon and low phosphorus, species 1 always wins. Low silicon, high phosphorus, species 2 always wins. Intermediate ratios, they can coexist. So high phosphorus, low silicon, right? Species 2 wins. Low silicon, low phosphorus, high silicon, species 1 wins. But in the intermediates, whether you're both down here or you're both up here, they can coexist. Remember, this is one of the answers to the paradox of the plankton. The idea that how can we have so many species out there when everything's well mixed? It's because species have different nutrients they can specialize to different degrees. And then we can also think of these complex interactions in microbial communities over time. So this is a diagram of a biofilm, perhaps something living on a rock in a stream or a lake. And if we start with a naked substrate, then these little things come in at first, the ones that press very tightly to the surface. They don't need to compete for nutrients. They don't need to compete for light because they're right on the surface here. And then over time, you essentially have this forest, this microbial forest that's coming up. And it'll actually create this sort of mucilage in here that holds everything together. And then they'll start competing for light. Now these organisms aren't doing so well down here because the nutrients can't get past these guys, and the light can't get past the stalk diatoms and the filamentous stuff. And then we can get some really big filaments, like Cladophora and such that we've seen, and they can overtop all this stuff. But they actually become this primary surface back here themselves, and so you can get another whole layer of epiphytes growing on the surface of them. And then in these systems, you could either have something like a flood that scrapes everything off, or a big old snail that comes and just takes the whole thing off. And the grazing pressure goes back. So these organisms and the snails can do okay here and scrape it off. And it gets too big for them, and they can't. Then you've got to have something that can actually pierce these things, because these are too big for the snails even to handle. Or some of the mayflies or some of the other grazing herbivores. Invertebrates. And then you get the flood, and you can go back and repeat that cycle. So this idea of succession and complex microbial interactions with each other is exemplified here, and how it relates to the rest of the community that's eating the microbial community. Okay. There are some examples of mutualism, facilitation, and then syntropy is a good example of that. And these are some of the things we talk about. Highly co-evolved mutualisms. Most of them are rare in freshwaters relative to marine systems. Anybody guess why that might be? So for example, we see, like, I don't know, cleaner fish in marine communities. We don't have an analog to that in freshwater communities. Or we see coral algal symbionts. We don't have as much of an analog to that, with it being dominant in freshwaters. What do you think it is about freshwaters that maybe doesn't allow them to develop as much?
Less space and less nutrients, so there's less niches to fill.
That's possible. Yeah, that's possible. I would say that's probably not the primary thing. What's one of the key ingredients we have for evolving diverse adaptations to the environment, where we see high degrees of diversity and endemism?
Necessity to all do it.
Well, what is it that leads to increases in diversity in an area? Time, right? So freshwaters are fairly ephemeral relative to marine. Marine has essentially been there since organisms evolved, right? But freshwaters have been there, but they've come and gone over time. So there's glaciations, there's changes in the tectonic plates. Watersheds can last for millions of years like the Mississippi, but there's less time for coevolution, and you need a long period of time for that to occur. That's not to say that there aren't any examples, but they're less common than other ones. So there are some examples of nitrogen-fixing symbiotic organisms. Having fun back there?
It's good.
Oh, the bee, yeah. So we know that in terrestrial systems, legumes are important and have nitrogen-fixing symbionts, but there are Azolla, there's ferns that have aquatic, that actually have cyanobacteria that live with them. Everybody's ducking down. Syntropy is another example. This goes back all the way back to the carbon cycle, when we talked about the complex series of fermentations and methanogenesis it takes to break down organic carbon to CO2. And it turns out that there's no single species that anoxically can take organic carbon all the way down to CO2. But if you get a complex assemblage of species, they are able to break it down. So if you remember, we had acetogenesis, we had methanogenesis, we had these simple fermentations. All these require different species. So this is not like aerobic respiration, where we get a situation where a single organism can take all organic carbon all the way down to and respire it all the way to CO2. Some images about mutualisms. In a sense, they're difficult to talk about in a general way, because organisms evolve to be mutualistic, help each other when they can do something for another organism that costs them almost nothing, that benefits the other organism, and the other organism can do the same back. And so that tends to be sort of context dependent and peculiar to life histories in the natural history of the organisms that are evolved. And so unlike, say, predator-prey interactions, which we'll talk about next, where everybody has to eat something and there's always somebody out there that's trying to eat you, you can get some real generalities there. It's more difficult with mutualism. But here's some examples. The first one is a nonstock species. It's a heterocystis cyanobacterium. And it is actually in fern Azolla. And Azolla has little pockets that in it that it sequesters this cyanobacterium in. And the cyanobacterium is able to fix nitrogen, and the Azolla just feeds it carbon basically, and gives it, so it doesn't have to photosynthesize, and collects nutrients for it. This is actually historically a very important symbiosis, because this is big part of the reason the Chinese were able to, and the Southeast Asians in general, were able to continue rice culture for many centuries without fertilizer, without extra fertilizer additions. So what we do to cropland and rice paddies now is use fertilizer that's been pulled out of the atmosphere, the nitrogen pulled out of the atmosphere, and the phosphorus has been mined, and we keep the productivity up and going that way. But historically, they would have these rice paddies, and they would basically set it up so that these organisms could grow and carpet the surface and fix nitrogen, make it nitrogen rich. And then they would drain them out and let the fern and the cyanobacteria essentially die and release their nutrients, reflood them and plant their rice. And the nitrogen would be there then to continue growing over many centuries, be able to use the same fields again and again. It's somewhat analogous to rotating soybeans in modern agriculture, when the soybeans have a nitrogen fixing legume, they're a legume as well. So that's actually something that's globally important in history. The next example here is nonstock, which is a cyanobacterium related to this one, but it has a midge in it. And this is also important mainly because I got some papers published on it, but almost nobody else really knows that much about it. This species of nostoc is found throughout the mountainous areas of Western North America for the most part, although it's been found in other places. And it occurs in low nitrogen areas, and there's a little midge larva that lives inside there. If you look very carefully, you can see its head right here and its tail right there. And when you go out to these systems, the nostoc that has the midge in it gets this ear-shaped look to it. So we're looking at it this way, flat, but if you turn it this way, it'd be quite thin. And perhaps have this little thicker area around it, just like your ear does, right, where the midge is inside there. The midge spends its entire juvenile life living inside of this cyanobacterium, and it eats basically nothing but either the nostoc itself, or there's a little hole it can stick its head out and clean the diatoms off the surface of it. The midge larvae actually secrete a silk, and they use it to lash these colonies onto the rocks that they're on. So when the floods come into these systems, the nostoc that don't have the midge all get washed away, whereas the ones that have the midge in them are able to withstand that force. In addition, this change in morphology that's driven by the midge from this spherical shape to the ear shape increases the surface area to volume ratio. And maybe you can guess where I'm going with this. What happens when you increase surface area to volume ratios? What's increased?
More relative diffusion?
Yeah, diffusion rates go up, and so they can basically get nutrients in faster, CO2 in faster for photosynthesis, and get rid of their extra oxygen and whatever else there is in there that they don't want to have. The nostoc does not help the nostoc to grow. In addition to that, when the silk is on here, it makes it so that these ears can stick out of the flow boundary layer further, and that also increases the diffusion rates as well. So it apparently stimulates that. And the last thing is that when this midge gets old enough to emerge and pupate, it actually causes the colony to fragment, and a bunch of bits go off, and they create a whole bunch of new colonies right when the adults go out and mate and the females are out to relay their eggs. Nobody's really got that bit where the females lay their eggs and they get into the colonies. But so that's just part of the biology that nobody, to my knowledge, understands yet. It is interesting in that you only have one of the midge larvae per colony, and if you put two together, they'll fight it to the death. Basically, one will win. But again, maybe there's only a limited amount of food there. So this is kind of a curious thing because you actually have a eukaryotic multicellular animal that lives inside of a prokaryote or a bacterium, basically. So it's a real turned around mutualism as far as anything that in the world. I don't think there's many of those. There's a lot of ciliates and animals that have algae growing inside of them, and they use that to be autotrophic at times and then provide them nutrients. So this is an example of Hydra. Your Hydra has these sticky arms with pneumatocysts on it, and it can get particles, and then it does that thing where, I don't know if you've seen them before, they actually cartwheel, where they kind of stick on their behind end and then they go down, and they can cartwheel around on solid substrates. if you put them in the dark, they'll get rid of this green alga. They'll expel it because they don't need the photosynthesis, and it's just a freeloader at that point. So, is this a mutualism? Maybe, if it protects the green alga from predation, or gives it more nutrients than it would get, but it also could be considered a conditional mutualism. This next example here is the diatom epithemia, and it's common in nitrogen-poor streams throughout the world. And epithemia has these little things here, are actually unicellular cyanobacteria. They're able to fix nitrogen. So it has cyanobacteria right inside of it that can fix nitrogen. As we talked about when we talked about nitrogen fixation, no eukaryotes that are known have nitrogen fixation, but they're able to have these little cells inside of them, and they basically just transform like chloroplasts. The cells divide, and as chloroplasts do, and their offspring get them. So this is another example of a mutualism. As you can see, a lot of these sort of revolve around nitrogen fixation. There are some other examples. There are some aquatic lichens, and remember, lichens are a mutualistic relationship between an alga and a fungi. There are some plants, some aquatic plants, submerged aquatic vegetation that has mycorrhizal interactions, as they have fungi attached to their roots that help them bring nutrients in. In terrestrial systems, they also help bring water in, but obviously aquatic, that's not going to be a problem. This is an example that we actually talked about before. This is a scanning electron micrograph. This is a heterocyst of a cyanobacteria, probably an Anabaena species. Photosynthetic cell, and these bacteria are all growing on the surface of that heterocyst. So the bacteria are getting a lot of carbon from the cyanobacterium, and this will increase their rate of respiration, which will decrease what?
O2.
Oxygen, right? And why would that matter to the cyanobacterium?
Because they don't want O2 production inside the heterocyst. I don't remember why.
Right. Because anybody remember why that was? They don't want O2 production inside there. What enzyme it inactivates?
Like nitrogenase? Or something?
Right, exactly. The nitrogenase, good. So the enzyme that takes nitrogen gas to ammonium nitrogenase is inactivated by oxygen. So you might remember we went through a series of adaptations that cyanobacteria had to reduce oxygen coming into heterocysts. So this then would be a mutualistic relationship between the bacteria that are fed by the cyanobacterium and the nonstock, the Anabaena, which can fix nitrogen more quickly in the presence of the bacteria. Okay, so that's the example of that. And just to there's a little bit of a rehash, and that is that most microbial interactions are mediated by chemicals, right? So I talked about a number of these symbioses I talked about were mediated by chemicals. We talked about attractions, allelochemicals, some of those things. Microorganisms, all organisms use chemical cues, but because microorganisms are unicellular, they don't have the ability to as easily do things like make an eye that can focus on something, or have larger organs that can sense changes in current velocities over distance or things like that. So they really get down to having to mostly rely upon the chemical characteristics of the environment. Now they can sense up and down, some of them, they can sense light and dark, some of them, but they can't necessarily, a unicellular organism can't form an image. So they can't distinguish between any particular object. So they use chemical cues, and there's some interesting research that shows that the sequence of phytoplankton that goes on in lakes, and we'll talk about seasonal succession, is actually stimulated by the chemicals given off by the previous organism that's there. So they can use them as cues for behavior, cues for timing of things, cues for protection from predation. And we'll see this in the larger organisms as well. One of the things that you need to do when you think about these interactions, you always need to consider the hydrodynamics. So if an organism is inside the diffusion boundary layer, they could have a strong effect on their neighbors, right? Because the diffusion flux rates are quite high in the close proximity there. But if you get out into turbulent flow, then there's a quick release. So we talked about interference competition, right? And the idea that an organism could maybe put out a chemical that would be toxic to another organism or harm it in some way. If you're a bacterium and you're interfering with your neighbor and you're in the flow diffusion inside the diffusion boundary layer, you're okay because it doesn't cost you too much to put that chemical compound out. But if the compound's getting washed away too quickly, then you can't, it's not worth your trouble to make that compound. In that case, those compounds you'd expect that are washed downstream, you'd expect them to be basically signaling compounds that an organism downstream is using to see what's going on in the world above them, essentially, or